Hello everyone, our topic for today is hypersensitivity. So you might be wondering what this hypersensitivity is. Yeah, it's all about your immune response. We all know immune response is actually a protective mechanism of our body. But now, when it gets excess, it becomes damage. It damages our own tissues, it causes diseases, or it even lead to death of the host. So that particularly is called as hypersensitivity. You may say this hypersensitivity as an immune response, and is generally immune response is generally a protective process, but it may sometimes be injurious to the host. Hypersensitivity refers to a condition in which the immune response results in excessive reaction leading to tissue damage, disease, or even death in the sensitized host. So, basically, we are classifying this hypersensitivity broadly into two categories. The first one is your immediate type, and the second one is your delayed type. In the immediate type, it occurs immediately and it recedes immediately. Whereas this delayed type, it only occurs after some 24 to 72 hours after the person is sensitized by the antigen, and it lasts longer. Immediate response is basically an antibody mediated immune response, humoral immune response, whereas this delayed type is cell mediated immune response, that is, it is based on T lymphocytes. And it transfers passively, whereas this do not transfer. This immediate response is short lived and it is easy, whereas this delayed type is difficult and it lasts longer. Immediate can be induced by any route, whereas this delayed is through direct contact. Now, let's see how Coombe and Gell classified this hypersensitivity. They classified hypersensitivity into four types. The type 1 is anaphylactic, and type 2 is cytotoxic, type 3 is immune complex, and type 4 is delayed. As we said, delayed belongs to the cell-mediated immune response type, whereas the rest 3 belongs to the immediate type, which goes to the humoral mediated, whereas this is antibody B-cell mediated. Let us discuss type 1 in detail. And before going to type 1, I forgot to tell something. Later, type 5 was also introduced, which is based on stimulatory. Now let's discuss type 1 in detail. This type 1 anaphylactic form can occur in two forms. First one is very fatal, that is anaphylaxis, and the second one is known as atopy, which is non-fatal. Let us see how the mechanism of this type 1 occurs. Before going to the mechanism, we have to get normalized with two terms. The first one is sensitization and the second one is the shocking. Okay, sensitization, sensitized dose and shocking dose. Sensitized dose is the first time an antigen, a person is encountering a particular antigen. Whereas the shocking dose is when the same individual is again attacked by the same antigen for the second time. So this is sensitization and that is shocking. Now I hope you understand the difference between what is sensitization and shocking. Let's move to the mechanism. When an sensitized individual is given a shocking dose of an antigen, this anaphylaxis is occurring. You may not understand this now, but let me explain you now. Okay. Sensitization, as I already said, sensitizing means the first time an individual is encountering an antigen. So this may occur through any route, but the most effective route is your parenteral route. Sensitization can occur through any route, but the most effective is your parenteral route. Okay. And there should be an interval of 2 to 3 weeks between your sensitization and your shocking, because within this 2 to 3 weeks IgE antibodies are produced in your body, and that helps in this anaphylaxis. Okay, An interval of 2 to 3 weeks between the sensor and its shock is required to produce this IgE antibodies. And this IgE antibodies attaches itself with mast cells and basophils through its Fc end. Okay, when a shocking dose of the antigen is given to that individual, this antigen is getting binded with the antibody, that is your IgE antibody. Now, an antigen-antibody complex is formed, and that releases certain chemical mediators which causes anaphylaxis. So, what is that chemical mediators? Again, these chemical mediators can be of two types, primary and secondary. Primary basically contains mast cells and basophil granules, whereas the secondary or newly formed mediators, that too it formed because of the stimulation through the mast cells, basophils and other leukocytes. Let us see these chemical mediators in detail. As I said, these primary mediators are histamine, serotonin and eosinophil chemotactic factor of anaphylaxis. So what histamine is? We know it is actually it causes vasodilation and it causes contraction of smooth muscles, and this is a most common human anaphylactic primary mediator. Okay, then serotonin is just opposite of histamine, which causes vasodilation, whereas the serotonin causes vasoconstriction. And eosinophilic chemotactic factor, these are some mast cells granule, and they contain certain chemotactic for eosinophils. They probably contribute to eosinophilia associated with hypersensitivity reaction. And now let us move to secondary mediators. And they include your platelet activating factor, prostaglandins, thromboxane, bradykinin, and slow reacting substances of anaphylaxis, etc. So, we have been discussing about anaphylaxis for this long. Let us know what are the features of this anaphylaxis. Yeah. Wait a minute. I'm coming. Features of this anaphylaxis. This anaphylaxis may take some few seconds to few minutes after the antigen is encountered into the host body. And this is mediated by IgE antibody, as we all know. And there are some organs that are getting affected because of this, and these organs are called as target organs, or they can also be called as shock organs, right? The most common shock organ is your lung that causes, it might end up in bronchospasm, laryngeal edema, and even death. Now let's discuss about the types of anaphylaxis. It can be anaphylaxis in vitro and cutaneous anaphylaxis. So this cutaneous anaphylaxis is basically literally little interesting, because now we are making use of this cutaneous anaphylaxis as a test dose for any sort of medicine. So I hope now you got to know what this is about. This cutaneous, the word cutaneous itself, it says about something related to your dermal layers. Yeah, this is given through intradermal. When an antigen is entering into your body intradermally, then this occurs. Cutaneous anaphylaxis may be induced when a shocking small dose of antigen is administered intradermal route to a sensitized host. It is manifested by appearance of local wheal or flare response. Cutaneous anaphylaxis is useful in testing for hypersensitivity and in identifying allergen responsible in atopy. Whereas this anaphylaxis in vitro is also called Sulzdane phenomenon, and this phenomenon is because of and this is literally a difficult one. When an isolated tissue of intestine or uterus of a pig is immersed in an ringer solution, the organ is undergoing contraction. The tissue is undergoing contraction, and this is known as anaphylaxis in vitro. I hope you understand the concept of anaphylaxis. In the next session, we will be discussing about atopy and other types of other type of hypersensitivity reaction. Thank you.